Good afternoon, everyone, and welcome to our monthly seminar series. We are extremely excited today to introduce Umar Bakali, our doctoral candidate from the Department of Biochemistry and Molecular Biology. He's going to talk to us a little bit about his work with our wildland firefighters and their exposure to polyaromatic hydrocarbons. Before we get started, we really want to just send out our thoughts and our prayers for all of our firefighters, families, and first responders that are working on the west coast of Florida. We hope you all stay safe, and please let us know if anything is needed from us during this time. So as I mentioned, Umar Bakali, he is a doctoral candidate pursuing his PhD in biochemistry and molecular biology in the laboratory of Dr. Sylvia Donner. His role in the FCI environmental sampling program is to analyze firefighters' exposure to toxic and carcinogenic compounds by using chemical extraction techniques and GCMS to isolate and quantify them. Specifically, he is looking at PAHs, PCBs, PBDEs, and PFA and PFAS. And so without further ado, I'm going to hand it over to Umar, and Umar, go ahead and take it away.
All right, appreciate it. Thank you, Natasha, for the introduction, and I echo the sentiments. We're really appreciative of our firefighters' work, and we hope you all stay safe on the West Coast as Hurricane Ian makes landfall. So what I have for you today is, like Natasha said, our work on really a pilot study for the occupational exposure in wildland firefighters to polyaromatic hydrocarbons. So really to start out, first, we're going to take a look at wildland firefighting in comparison to structural firefighting, which has really kind of been the bread and butter for the FCI thus far. What we've really looked at is structural firefighting and just showing how wildland firefighting can really be a totally different animal in comparison. And we'll go through the study design and the methods that we use for monitoring the exposure, which is quite similar to what we've used in the past, and finally the results. So first, the background. So when most people think about structural firefighters, or rather just firefighters in general, they think about these guys here. So this is a photo taken by Dr. Emery Dikichi in Sylvia Donner's lab. And when we went to West Palm Beach up with Chief Nimchek to observe a Class B controlled live fire training. So when you look at a structural firefighter, you see you have this really heavy turnout gear, bunker gear that's, you got a liner inside. It's very, very heavy protection. You got the SCBA mask, you got contained air, and you have evidently usually pretty good access to water. In this case, they're mixing it with PFAS to create a foam for a Class B fuel. So structural firefighters are who you see in cities and towns. They'll be fighting fires inside buildings, inside structures, if it's a car on the road. And generally, most of our exposure research and research on firefighting in general has focused on structural firefighters' exposures. But wildland firefighting is comparatively quite different. So to look at wildland firefighters, this is a picture of some of them in Lake Wales Ridge State Forest up in Lakeland. They're dressed a lot more lightly. So fire-resistant turnout jackets, no liner in them. They got a helmet. They're using lighter vehicles, ATVs, UTVs. They've got hand tools, chainsaws, things like that. But the most striking difference is that they usually don't have any respiratory protection whatsoever. Maybe they have like a gator mask, if at best. And according to Navarro et al., Kat Navarro, she's a wildland firefighter herself, as well as a scientist who I had the pleasure of meeting early on in the process when we were first discussing the mechanics for our pilot studies for wildland firefighters, noted in a paper that wildland firefighters are estimated to be up to 43% more likely to develop lung cancer compared to the general population, which is more than the general population and actually more than structural firefighters are. So, and this is mainly due to their lack of respiratory protection. So there's a few more intricacies of wildland firefighting. Compared to structural firefighting, burn areas are much, much larger. So structural fires are a matter of square feet, maybe a block if it's a really bad fire. But wildland fires are a matter of acres, a very, very vast area. Limited access to water, like I said before, and structural fires may last for several hours, but wildland fires may last for many days. And of course, the first thing when I arrived on scene at Lake Wales Ridge State Forest, the first thing that the burn boss told me, Dan Flint, ready to eat some smoke. Work tasks are too strenuous to be using respirator filters. So smoke inhalation is a foregone conclusion when you're a wildland firefighter. A couple of other occupational health concerns that I'll touch upon, but I won't really go into in this presentation, is that wildland firefighters don't really often get the opportunity to launder their PPE. So what they get exposed to during one fire event may stay with their PPE and when they go to the next one, and it'll stay there and it'll rub off onto their skin, and it can cause some damage in the long run. Some other things I noticed from speaking with wildland firefighters at these sites, the presence of illegally dumped materials may also affect what a wildland firefighter is exposed to. So it's not just foliage and forests and grasses that are burning, but instead, if someone decided to illegally dump paint and tires and couches and things like that in the middle of the forest, and it has happened before, a wildland firefighter could be exposed to the byproducts of those materials burning as well. Also, some other interesting things about vegetation that's burning. So poison ivy, normally if you rub up on it and the urushiol get onto you, that'll cause an allergic reaction and cause you some harm. But when those plants burn, the poisonous compounds can also be volatilized and be breathed in by wildland firefighters. And just in general, some vegetation may burn more vigorously or produce more smoke. So for example, I heard about the invasive plant species like Kogan grass and kudzu, for example. Those, when they burn, produce very thick black smoke, which may have a different composition to it compared to the rest of the environment. And lastly, what I found very interesting was the use of herbicides for treating actually these invasive species. If an area catches fire that was recently treated with herbicides, the fumes of these herbicides can come into the air and also expose wildland firefighters. So just some things to take a note of there. So really the bread and butter of our pilot studies was through prescribed burns. And prescribed burns are like a simulation of wildland fires and yet are somewhat different. They're done for a couple reasons. They're done to reduce the accumulated fuel on the floor of a forest, so that there's less fuel for a prospective wildland fire to burn. And they're also used to maintain fire-dependent ecosystems, especially the flatwood areas around the Gulf Coast and in the Southeast. So these ecosystems, especially the ones I saw in Pine Log State Forest and in Lake Wales, have a lot of pine trees in them and generally have a relatively vacant understory. So it's just grasses and shrubs, and things like that. But if left alone with no fire, what will eventually happen is that other species will kind of take advantage of these flatwood areas and take root. And so burning, what that allows, what the service that this provides to the ecosystem is essentially it takes care of those invasive species, returns the nutrients back into the soil, and helps these pine trees and the ecosystem to maintain itself. So compared to wildfires, prescribed burns are much more controlled. Obviously, you don't want to set a fire during a situation where you can't control it, especially in the wildland. So for example, some things to take a note of, they specify an area, a certain acreage or tract of land before burning it, of course. They want target weather conditions, not too windy, lest the fire go out of control. Obviously, can't set a fire during the rain. Want to also make sure that the heat index is okay, lest you have wildland firefighters getting heat exhaustion. So the caveats, what makes this a little bit different from a true wildland fire is that these prescribed burns are performed under a shorter time frame. So these will be, for a few hours or for most of the day, but they won't hopefully go for more than a day. And so the higher degree of control may not necessarily be representative of an actual wildfire, especially when it comes to smoke exposure. When combating a prescribed burn, wildland firefighters will try their best to not be downwind of the smoke if they can avoid it. But during an actual wildfire, all the bets are off. You do what you need to do. So here on the right, this is an example of a wildland firefighter using a drip torch to dispense burning fuel of a ratio of gasoline and diesel that kind of sticks around and produces a perimeter from which the fire burns inward. So as with most fires in which carbon-containing fuel is the subject that's burning, polyaromatic hydrocarbons tend to be the most ubiquitous carcinogen around. So these are compounds that are produced during incomplete combustion. So any situation where you have a fuel-rich and oxygen-poor situation. So unlike a structural fire where you get oxygen poor very, very quickly, the oxygen inside a structure gets eaten up very quickly, a wildland fire may have more oxygen available to it just by virtue of it being outside, but you'll still end up producing PAHs simply because of how quickly fires will gobble up the fuel. So these compounds are interesting, and they can be used as proxies for carcinogenic exposure monitoring, or really they are carcinogens in and of themselves, IR group 1 and 2b, because they can act like particles or they can act like vapor in their deposition, depending on the size and the temperature. So lighter molecular weight PAHs, the 2-ringed ones like naphthalene, will be more in the vapor phase in ambient temperatures, and especially in the higher temperatures, whereas you'll need higher temperatures to vaporize and volatilize the heavier ringed PAHs like this one here, chlorine. And when you have very high temperature situations for prolonged periods of time, eventually you get ash and soot from these PAHs coalescing on each other, and that will essentially give you the ash and soot that ends up depositing on gear. And as a 1-2 punch, on top of these particulates, you can also have PAHs that kind of hitch a ride. So that's how, even in ambient temperatures, you can still have exposure to the less volatile PAHs as long as you have particulate matter around that can drift. So the routes of PAH exposure are threefold. There's 2 of them that are more salient to wildland firefighters. So there's inhalation, ingestion, and deposition as far as exposure goes. Inhalation being, of course, you inhale the compounds, whether it's the particulate matter or whether it's the volatilized PAHs. Ingestion, if some of it falls on your food or in your drink when you're drinking something. And then deposition when it gets on your skin, and then it stays there long enough and hasn't been washed off, so it can permeate through your skin. Once the PAHs are internalized, the carcinogenicity comes from the metabolic pathway that's used to get rid of it. So when you have an kind of like an unknown compound inside your body, the body tries to get rid of it by excreting it. And because PAHs are so hydrophobic, meaning they don't dissolve in water and they won't really come out very well in urine, they will to an extent, but not a lot. What the body tries to do is use xenobiotic metabolism to essentially make these compounds dissolve in water. The issue with this is of course, that this produces a very reactive compound. So for example, this pathway for benzoate-pyrene, which is a group 1 carcinogen, produces the diol epoxide, which is very reactive with DNA and can produce adducts, which leads to dysfunction of transcription or replication pathways, and ultimately can lead to carcinogenesis if it happens 1 too many times. PAHs can also disrupt apoptotic pathways. So in the case that a cell becomes carcinogenic, it doesn't kill itself. So, and that can result in delays in tissue repair and also carcinogenesis, and also increase inflammation, which can adversely affect other conditions outside of cancer. So let's go through now with our study design, the aims and techniques with which we use to evaluate wildland firefighters' exposures here. So our study design and aims were threefold. We wanted to first document occupational factors that influence exposure. So the length of the fire event, how long did wildland firefighters stay in the fire? What was the acreage of the fire? How big was it? What sort of occupational tasks were performed? So were they a sawyer? Were they ignition? What instruments did they use? And then of course, terrain and burn characteristics. What was the terrain like? What sorts of fuels were burning, and so on and so forth. And then we wanted to take a look at ambient exposure through the air by passively sampling using silicone passive samplers. It's something that if you've tuned into previous seminars with the FCI and ESP, that you're probably very aware about silicone wristbands. And then lastly, we wanted to determine the gaps in a wildland firefighter's PPE. Can we determine which areas of the body are most likely to be affected by carcinogenic exposure? Like, where does it go the most? Using alcohol swabs for dermal swabbing. So starting out, let's go through with silicone wristbands as passive samplers, what we've used in the past. They're very, very effective as passive sampling devices for volatile compounds because of how rugged they are. They're very, very temperature resistant as long as you're not directly inside a fire. They won't burn. They're very cost effective, very inexpensive. Hardly anything is more invasive than, or less invasive, I should say, than just wearing a silicone wristband. And so really they present an ideal means for monitoring carcinogens in firefighters, and in this case, wildland firefighters. And the way they work is by using the very porous microstructure in the silicone that can actually serve to uptake the volatiles into the matrix. So following that, after they've been prepared by solvent cleaning and vacuum oven, going through a vacuum oven, to pre-clean them before being deployed to the wildland firefighter to wear, they're solvent extracted. So all the analytes, all the compounds that we're looking for in there, in the silicone matrix, can be extracted off the wristbands, and then that can then be pre-concentrated for analysis. In regards to dermal sampling, we know that wildland firefighter PPE, both in the Florida Forest Service and in the US Forest Service as well, is fairly standardized. Generally, when you look up a picture of a wildland firefighter in the United States, you'll see that they usually have something like a yellow turnout jacket made of Nomex and a green set of Nomex pants, a helmet, usually a line pack as well. So aside from minor little differences between, one set of wildland firefighters and another, or, what's necessary for one job task versus another, the overall gear is mostly the same. So we wanted to check and see what sort of contamination intrusion occurs through wildland firefighter PPE. And to do that, we had wildland firefighters swab themselves using alcohol swabs, just clean room grade isopropyl alcohol wipes with nothing else added onto them. So sort of reminiscent of a decontamination wipe, but just isopropyl alcohol instead of some other sort of detergent. So we had them swab once on each side of the neck, once at the base of the neck in the thyroid region, once for each arm, once for each inguinal fold in the groin area, and then one for each leg. So a total of 9 swab samples, each of them collected in individual airtight foil bags. And then once we got back to the lab, we were able to extract them and then analyze them. So our analytical methodology is gas chromatography and mass spectrometry. It's a methodology that's used also by fire investigators and forensics experts in order to determine the sorts of accelerants in an arson, for example. We use them for determining the presence of the 16 EPA priority PAHs. So we operate this based on the EPA 8270 series methods for semi-volatiles via GCMS. And the way that it works essentially is you have your extract, it gets injected into the sample injector where it's heated very, very quickly. So it vaporizes almost instantaneously. It's then condensed onto a column, which acts like an obstacle course for each of the different compounds inside that extract. And then one by one, they'll go through the column and will be fragmented using electron ionization to be read by a mass spec. So what this essentially gives us is a combination of a retention time, so the time that it came out of the column, and the fingerprint, so the mass spectra of the actual compound. So this allows us to both identify and quantitate, if we use it with calibration curve, what compounds were there and how much were there. So let's take a look at our results. But just before we do that, let's go through the story of the 2 prescribed burns that we went through. I know I've gone through a lot of background already, but this is kind of germane to the overall results. So we went to 2 prescribed burns with lots of similarities and some differences as well. The first in Pine Log State Forest in June 15th and 16th, and the second at Lake Wales Ridge State Forest on June 23rd. And so the first one in Pine Log State Forest had 10 wildland firefighter participants. It was a comparatively small burn area of around 47 acres. And the length of the fire event was approximately 2.5 hours. So started around 9:30 in the morning and finished by noon. In Lake Wales, we had 13 wildland firefighter participants, and the burn area was much, much larger, nearly an order of magnitude, so about 430 acres area. And the length of the fire event lasted for approximately 8 hours. But these two burns overall were pretty similar in the fact that they had roughly the same type of terrain and the same type of vegetation around there. So we could expect the overall emission profile for the PAHs to be roughly the same. The ambient conditions at these two burns, so on the left, again, Pine Log State Forest, and on the right, Lake Wales, were roughly the same as well. Relative humidity, 47%, 40% for Lake Wales, around 37 degrees Celsius ambient temperature, 98 Fahrenheit for Lake Wales and Pine Log State Forest both, and then not too much of a difference in wind speed and direction either. And this is really representative of the type of weather conditions you would want in the first place to be conducting a prescribed burn. Albeit you could argue that these are actually a little bit hot, and in terms of the heat index, it might be still a little bit on the risky side for wildland firefighters. So comparing the personnel distribution, so what wildland firefighters took which tasks. They're not exclusive, so participants could indicate more than one task. The majority of them were involved with ignition. So like I showed before, using a drip torch to chop fire around the perimeter of the prescribed burn. We found that the use of ATVs and UTVs was more so in Pine Log State Forest. It was really about the same, actually. 6 out of 10 in Pine Log State Forest and 4 out of 13 in Lake Wales. So generally ATVs and UTVs are used fairly frequently, but brush trucks weren't used at all, actually, or not indicated so in Pine Log State Forest. And I would imagine that this is just because the size of the prescribed burn at Lake Wales was so much larger, so it actually necessitated the use of brush trucks to carry instruments. So let's take a look at the comparison between a baseline measurement for the wristbands for wildland firefighters at Pine Log State Forest compared to wristbands worn during the prescribed burn. So the day before we had the prescribed burn at Pine Log State Forest, we gave the wildland firefighters wristbands to wear over a period of 24 hours. So given to them at 9 a.m. the day before the burn, and then collected 9 a.m. on the day of the burn before the burn happened. And by comparison, these are the wristbands that were worn for a period of about 2.5 hours and collected after the prescribed burn. And we can see that compared to the baseline, the average exposure during the prescribed burns is about 5.2 times higher than the baseline in really only a tenth as much time. Albeit a lot of the exposure comes from this one single firefighter who happened to get a lot of exposure. And I'm quite suspicious of this one. I'll tell you a little bit more later. So for most of my statistics afterwards, I discounted this outlier. So not counting this outlier value, prescribed burns in only about a tenth as much time, we had almost 2.5 times more overall exposure. By comparison, the wristbands collected from Lake Wales Ridge State Forest, as you might expect from it being such just a much larger burn and for a much longer period of time, had much greater exposure, somewhere around the order of 3 to 4 times more than the prescribed burn at Pine Log State Forest. Unfortunately, we weren't able to collect baseline wristbands for this one because of the logistical constraints we had. So the funny story behind this, Dr. Caban had told me about this burn. He had gotten into contact about this burn, and I was having lunch literally the day before the burn was happening. So 9 p.m. that night, I was up at the hotel at Lake Wales, and the next morning we were at the burn site. So unfortunately, we weren't able to get a baseline measurement. But comparing these two prescribed burns side by side, we can see that generally the overall pH exposure is mostly from lighter pHs, so those from naphthalene down to pyrene. And then we see a pretty sharp drop off afterwards. We see some heavy pHs here and there, but not really much more than the lower limit of quantitation for our method. And we definitely see more in Lake Wales, but again, a quite apparent drop off between the lighter molecular weight pHs and the heavier molecular ones. What we also see is some bands that would tell us about the emission profile of the types of fuels that we're burning. So especially for naphthalene, phenanthrene, and pyrene, you can see these bands are a little bit pronounced here and then greatly pronounced for Lake Wales Ridge State Forest. So it gives us a little idea of similarities in fuel and burning vegetation. So it's easy to say that pH exposure is greater at larger burns, but is that the end of the story? And for me, I thought the answer was no. What I did was decided to take a look at time-weighted average pH exposure. So normalizing the exposure that the wildland firefighters experienced by time, by the amount of hours they spent. So 2.5 hours for Pine Log State Forest and 8 hours for Lake Wales Ridge State Forest. And then I also stratified them into the lighter molecular weight, heavier molecular weight, and then total 16 pH groups. And what we found was that, surprisingly, the overall exposure to pHs seemed to be quite similar if you normalize per hour at both of these controlled burns, despite the fact that Lake Wales Ridge State Forest is so much larger, or at least the prescribed burn there was so much larger. And so what we could conclude from just looking at these two burns is that the time spent at these burns is likely the major driving factor for wildland firefighters' exposures. Well, why is this important in the first place? Well, so wildland firefighting is a profession where you have hazards with very few controls. You can't eliminate wildland fires, or if you could, there wouldn't be any wildland firefighting. So the point is moot. It's hard to substitute them too. And really the exposures that you get in wildland firefighting are only partially mitigated by PPE, and especially by necessity of not being able to use the respirator masks and SCBA that a structural firefighter could use. So one opportunity for exposure control in wildland firefighting is really just reducing a wildland firefighter's time at the scene, whether that means rotating out wildland firefighters during a prescribed burn, or perhaps or some other method. And then when we take a look at pH intrusion using alcohol swabs, so this is where we took a look at the gaps in the armor, so to speak, for the wildland firefighters' PPE, what we found most strikingly was that despite having the lowest surface area, the neck had some of the highest rates of exposure, or the highest overall exposure. And the way we measured this was essentially extracting the swabs and then converting based on the volume and the concentration that we found to the total nanograms of total pHs that we found on each swab. And then averaging them in the case that summing them up in the case where we had two swabs. So two swabs to the neck, those were summed together and then averaged for each of the participants. So the neck definitely had the greatest exposures despite being a really low surface area sort of anatomical area. But overall, what we did see was that with the larger burn in Lake Wales, we saw more exposure compared to the smaller burn in Pine Log. And in all cases, we saw more exposure compared to the baseline. What is a little striking to me, though, in the arms category is that the average appears to be much larger for Pine Log State Forest. And that comes back to the single participant that I mentioned earlier who had all that exposure. It turns out that one of the swabs, the swab for one arm had a lot of exposure, but the swab for his other arm didn't have much exposure at all. It reflected really the average that you'd see on everyone else's. So that also accounts for the large amount of variability here. So I suspect that this wildland firefighter was trying to help the cause of the study and specifically kind of dirtied up his arm a little bit. So in a nutshell, what would I like you all to take home from this? Generally, the majority of exposure to pHs observed is from more volatile, lower molecular weight compounds. But of course, the wristbands that we use are a little bit biased to looking towards lower molecular weight, more volatilized compounds rather than particulate matter. So using sampling methods that are more tuned for particulate matter capture may give us a more clear picture as to firefighters' exposure to both particulate matter and to heavier molecular weight pH contamination. And just in general, the presence of the volatiles and particulates are significantly impacting wildland firefighters' respiratory health in the first place. Time at the fire scene is the main driver of exposure, but terrain and fuel composition, I would imagine, if we did further studies, may also have impacts. So what effect would other sorts of vegetation have in terms of influencing wildland firefighters' exposures? And then the neck area is the most significant gap in wildland firefighters' PPE, likely because it's the area where there's the most air circulation going on. Everywhere else, the firefighters' sleeves are connected to the gloves. The firefighters, the brims of their pants, like tucked into their shoes, and then their jackets tucked into their pants. It really just isn't as conducive to pH intrusion as the rest of the neck is. So before I end, I'd like to give a few acknowledgements, especially to Dr. Sylvia Downer, Dr. Alberto Caban Martinez, Dr. Sapna Deo, and Dr. Natasha Schaefer-Sali for their guidance and mentorship on this pilot study. And then also the members of our lab, so Dr. Amrita Kichi, especially for the photos and for some guidance, and my lab colleagues as well, Alexia, Chitvan, and Marshley for their help with advice on the samples. And Chitvan actually went with me to one of these burns to Pine Log State Forest. Marshley helped me with some of the analysis as well. A lot of our research assistants on the Caban lab side. So Megal also went with me. Nishal was absolutely instrumental for helping me analyze hundreds of these samples. And then Ian Lee and Victoria Gomez as well for their help with the data entry. And especially big thank you to the Florida Fire Service, starting with the leadership. So State Safety Officer Larry Grubbs for helping us arrange these meetings with the various districts throughout Florida. So and Chief John Rollerson as well, and the district managers Mike Mathis and Pat Keogh at Chipola and Lakeland, respectively, for having us over to observe these prescribed burns. And then the burn bosses at Pine Log, Don Showalter and Dan Flint at Lake Wales, for kind of serving as the liaisons for our research team. And of course, all the wildland firefighters who participated in this study. And I'd also like to mention that this research was supported by a pilot project training grant from the Deep South Center for Occupational Health and Safety at the University of Alabama at Birmingham, and by the state of Florida appropriation to the University of Miami Sylvester Comprehensive Cancer Center for the Firefighter Cancer Initiative. And with that, I'll end. Thank you all for your attention, and I'll field any questions now.
Awesome. Great presentation, Umar. Thank you so much for sharing that.
Absolutely.
So I do see a couple questions coming through. Not sure I missed it. Would volumetric assessment be useful or feasible to address the questions on PAHs? Why or why not?
So I'm having a little trouble of understanding the question. So what would volumetric mean exactly in this context? If you mean specifically like the amount of like volume of air per time. Okay, there we go. I do believe so, especially in regards to particulate matter contamination. With regards to passive sampling, though, for volatile collection, the way that those passive samplers work is essentially you'll have a curve of volatiles that will absorb onto the polymer of the wristband until it reaches equilibrium. And after that equilibrium is hit, presumably after many, many hours or many, many days, you would then like not really see a change in concentration. But as long as you're up in that curve where volatiles are still absorbing onto the pH and not dispersing out, you would end up having a fairly accurate snapshot of what a wildland firefighter was exposed to. So volumetric assessment would be good for specific types of active sampling rather than passive sampling. So, okay.
Great.
Yep.
Next question we have is, could the accelerant contribute to the findings?
So to an extent, so the accelerant in this case would be the safety fuel that they would use. So the fuel, well, it's called safety fuel in the context of structural firefighting. I don't think they actually call it that in wildland firefighting, but the fuel they use is diesel and gasoline mixed, and either a 2-to-1 or a 1-to-1 ratio. I could remember. I imagine that fuel burns off very quickly, and the rest of what we're seeing is mainly just the foliage and the forest itself that's burning. So if it does contribute, the effects would be very, very minor.
Great. Thanks for sharing that.
Mhm.
So we have another comment. Very nice talk on your pilot study. Have you thought of correcting your alcohol wipe pH data for area swab to give you mass per unit area? Have you thought of using personal breathing zone sampler to assess inhalation exposure to compare to the skin exposure?
So, okay, great. These are two very good questions. So yes, when I did look at the alcohol wipe pH data, I was very aware of the fact that I really should have looked at the mass per unit area as well. Unfortunately, when working with several wildland firefighters, and things can get a little bit tough to kind of demarcate with them when it's just one or two of you and it's like a room full of wildland firefighters. So you can only really get so much information as to what exact area was swabbed. And then as for using a personal breathing zone sampler, that's actually one of my goals for future work. I would like to see through the use of exhaled breath monitoring. So for example, using exhaled breath condensate to see what exact compounds come out and how much from firefighters' exhalations post a prescribed burn or post wildfire. The logistics of that are a little bit tough because, exhaled breath, these volatiles will clear out pretty quickly. And in the case of a true wildland fire, these samples might be tough to collect and then preserve on their way back to the lab. But those both of those are two areas of improvement for the research for sure.
And that's a great segue for your next question. So it says, thank you, Umer. Great work and presentation. Perhaps you could elaborate on what you plan to do in the future for the audience so they see where we are going, given the importance of wildfires and how they are increasing because of climate change. So can you elaborate a little bit more about what your plans are for wildland firefighters?
Yeah, absolutely. So there's definitely a lot of different ways to take this research to different pathways. One of them is just by expanding the compounds that we're looking for. So not just PAHs, but also, for example, the herbicides that I discussed before, right? After a area has been treated and then that area gets caught on fire, what sort of, do we see any herbicides or byproducts of those herbicides in the air, and how much of it is there? Or then besides increasing our library of toxins to look for, what about internalized compounds? So like I said before, about looking at exhaled breath to see which compounds are internalized and how much of them are there. So like the BTEX compounds, for example. We could also look at in terms of using urine samples to see, compare how exactly post a prescribed burn, how those exposures correlate on the outside compared to what's internalized. So there's definitely a lot of different routes to go that way.
Great. We're looking forward to see what you do.
Mm-hmm.
Would there be any utility for real-time assessment of pHs linked to physiological biomarkers?
I think so, yes. Real-time assessment of pHs is actually a project that's done by a colleague of mine, Shadvan, using solid-state sensors. So by comparing a firefighter's exposure to pHs in real time, and then taking physiological samples to take a look at, perhaps the emergence or regulation of inflammatory biomarkers, that could be an interesting area of study, especially for looking at non-carcinogenic related illnesses, or non-cancer illnesses rather. So things that are related to inflammation, how they impact other comorbidities.
That's great. Oh, we have another, Dr. Donner is able to join us and, well, she's been on, but she's able to ask a question live. Go ahead, Dr. Donner. Sylvia, your hand's up. Are you there?
Oh, sorry, Natasha. No, I forgot to put it down.
Oh, okay. No
it
problem.
was the question that I asked
You typed
Umer
in.
to expand on the futures for the audience. So anyway, but thank you, Umer, for clarifying and explaining where we're going in the future.
Absolutely.
Great. All right. So thank you, Umer, again. This was another fabulous seminar for FCI. We will be having our next seminar series next month. I will be presenting, myself and my colleague, Dr. Chris Widlich, from School of Nursing and Health Sciences. We will be discussing mental health and specific to our USAR firefighters. And so with the timing of hurricane season, it is definitely timely that we are talking about our USAR firefighters. And again, we are thinking about all of our firefighters on the west coast of Florida, all of the departments, all of your families, and then also we know our USAR firefighters who are deployed over to the west coast who may not live there. So thank you guys for your service. Please stay safe, and we look forward to seeing you all next month. Take care.
Bye, everyone.